Well, hi, everybody. Uh, Welcome on this beautiful spring day uh, to our annual Breakthrough Challenge Breakthroughs in Action. I'm Bob Vonderheid. I'm director of the Abramson Cancer Center, and our incredible gratitude to all of you who support and participate in the Breakthrough Challenge um, and partnering with the Abramson Cancer Center. As you know, it's one of the most special partnerships we have. And thanks for joining today. We have some great, exciting updates from the scientists and the clinicians who are um, benefiting from the money raised by Breakthrough Challenge, and we're going to have a good conversation. Um, I just wanted to say a few things at the start. Um, it has been amazing for me how Breakthrough Challenge has been able to keep going and accelerate during the pandemic, and at a time when we were worried about losing connectivity with each other in terms of the scientists and the clinicians being able to be together physically. Um, Breakthrough Challenge has been something that's really been uniting and been fostering collaborations. And so this has really been an important avenue and mechanism for the Abramson Cancer Center. And I'm really proud about it. And I tell everybody I meet about how we're getting it done. Special thank you to the Breakthrough Challenge board. You all are amazing. And your confidence in us to find discoveries is inspiring, and we're here to do our best. I want to just recap a few of the statistics for Breakthrough Challenge. No longer Breakthrough Bike Challenge because we do more than bike. Raised now more than $1.9 million since the first ride almost 10 years ago, and 100% of the money raised by the participants goes directly to research here at the Abramson Cancer Center, and we've pulled out 4 projects, most recently funded, for you to hear about today. But more than just the 4 today, keep in mind that 43 faculty have received grants, and through this grant mechanism, we designate them Buzz Cooper Scholars. Buzz Cooper had my job a few years ago, and we're really pleased to have this opportunity to honor and remember his great work for the Abramson Cancer Center. So keep in mind that we're having the event again this year. September 18th, Sunday, is the in-person bike ride at the Daniel Boone Homestead, where we've been before. And in addition, there'll be a 2-week virtual event for spinners, runners, walkers, cyclists, and that starts September 5th, and will be sort of our launch into the main event on September 18th. So we're hoping everyone can join us for virtual activities and certainly the in-person activity on Sunday, September 18th. I keep saying the date so that you can write it down and make sure it's clear on your calendars. It's going to be a great year because we are going to exceed the $2 million mark donated by Breakthrough Challenge. And so I think we're all going to be super excited to be there. So, okay, without further ado, and so many people tuned in, this is great today. We'll jump right in. And as I mentioned, we have 4 presentations. Every scientist who you'll meet today is a recent recipient of a grant from the Breakthrough Bike Challenge. Just so you know, scientists apply. We send out a request for applications, and then all the proposals are vetted by colleagues here at Penn, very much like an NIH study section, and we identify the most promising ideas and able to award them sizable amount of money to really launch and accelerate and make their ideas and make the discoveries. So our first presenter is Anne-Marie McCarthy. Dr. McCarthy is Assistant Professor of Epidemiology in the Department of Biostatistics, Epidemiology, and Bioinformatics. And she's going to tell us about using a combination of genetic and radiology markers to identify folks at risk for breast cancer. So Anne-Marie, welcome.
Wonderful. Thank you so much, Dr. Von der Heide, for your introduction. Can you see my screen?
Yes.
Wonderful. Well, I'm so honored to be here today to present our ACC-funded project to you on behalf of our multidisciplinary research team. Breast cancer disparities are a huge problem in the US and in Philadelphia. This figure shows breast cancer mortality rates over the past 40 years or so in the US. While you can see breast cancer mortality rates have decreased over the past few decades, Black women in the US have higher breast cancer mortality than white women. So Black women here are the top pink line and white women. So you see this persistent disparity that hasn't narrowed over time. And in Philadelphia, Black women have 50% higher breast cancer mortality than white women, despite high rates of mammography screening. So we need to do something different to improve breast cancer outcomes for Black women. And the goal of our project is to use novel imaging biomarkers and genetics to improve early detection for Black women. So breast density is one of the strongest risk factors for breast cancer that we know of. Breast density is the relative amount of fibroglandular tissue versus fat tissue in the breast. And so here up in the top left corner, you can see an image from a mammogram, and you see the white tissue, which is the dense fibroglandular tissue relative to the darker fat tissue. And so women with dense breasts have 3 to 5 times higher risk of breast cancer than women with less dense breasts. And so this is a very useful risk marker. At Penn, we're going a step beyond breast density. We're using radiomic markers. And so my co-PI, Despina Cantos, is a computer scientist, and she's developing computational methods to examine breast tissue called radiomics. Her lab uses machine learning and AI to identify patterns in the breast that are linked to developing breast cancer. And we hope that we can use this to really pick out the women who are at highest risk so that they might choose to do more intensive screening. In addition to radiomic markers that we're developing, genetic markers can also help with breast cancer risk assessment. And so you've probably heard of BRCA1 and 2 genes. These are genes that drastically increase risk of breast cancer and ovarian cancer as well. And while they increase risk a great deal, they're also very rare. So what we're focusing on here in our project is common genetic variants. So differences in DNA between people that can also increase breast cancer risk. But each genomic variant increases risk only a small amount. And so what we do is we develop these polygenic risk scores, which summarize risk based on common genetic variants across the genome. And so for breast cancer, we have a 313 variant polygenic risk score that's been developed that can identify women with high, moderate, or low genetic risk for breast cancer. And so while BRCA1 and 2 mutations are very rare and only a few women will test positive, everyone can measure their polygenic risk score to see where they fall in terms of. So in our project, we plan to bring together radiomic markers and polygenic risk scores in order to identify which women are at highest risk of developing breast cancer so that these women might consider more intensive screening. Now, most of what we know about breast cancer risk is really based on studies of white women. And so in our project, we're specifically focusing on Black women because we want to understand how well radiomic markers and genetics can predict risk for Black women. We're uniquely positioned to do this work at Penn because we have a very diverse patient population. We have the ability to use electronic health records to build study cohorts, and because we have the Penn Medicine Biobank, which is a large ongoing initiative to obtain DNA samples from our entire Penn Medicine patient population. And so the ultimate goal of our project is to develop and inform precision screening. So what is precision screening and what might that look like in the future? So precision screening involves using a person's individual risk to determine when to start screening, how often to get screened, and what type of screening to use. So let's consider a hypothetical patient named Ms. Franklin. Ms. Franklin is a 42-year-old Black woman with no family history of breast cancer, and she's trying to decide whether to start breast cancer screening. So with precision screening, we would give Ms. Franklin a genetic test And based on her genetic test, we see that she has a high polygenic risk score. So based on that high polygenic risk score, she decides to go ahead and get her first mammogram right away at 42. When she gets her first mammogram at PCAM, she's found to have dense breasts. So we then measure the radiomic markers using Dr. Cantos's automated algorithm, and her radiomic markers also indicate high risk. So then based on her high risk based on the polygenic risk score and radiomic markers, we would refer Ms. Franklin to get breast MRI screening, which would be covered under insurance, under her insurance for high-risk women based on a new Pennsylvania law recently passed. And so Ms. Franklin would then be counseled to come back every year for both mammography and breast MRI to improve early detection of breast cancer. And we believe that more accurate risk estimates coupled with precision screening could help reduce breast cancer disparities and improve outcomes for Black women.
Thanks, Anne-Marie. We're going to have an opportunity for questions, particularly from the audience, after we hear from all four. But let me just ask you a clarifying question. First of all, we played a small part in lobbying for that state law that you referred to passed, thinking that once the law was there, then now we could do a research project like this. So it's all coming together. Can women get the polygenic risk score testing done now? Is this something that's already available?
That's a good point to clarify. So typically for the general population, PRS tests are not available. It's more in the experimental phase. There are some testing companies that are offering polygenic risk score tests for breast cancer, but it's primarily in the setting of women who are getting genetic testing for high-risk mutations like BRCA as well. It tends to be an add-on test. Right now, polygenic risk score tests are not covered by insurance. So we're hoping to generate the evidence that will help support implementation of polygenic risk scores.
Yeah, got it. That's great. Okay, thanks, Anne-Marie. And stand by. We're going to come back in a few. But first, we're going to hear from the rest of the speakers. And our next speaker is Erica Carpenter. Erica, there you are, Research Assistant Professor of Medicine. She's also going to tell us about combining genetics and imaging, but slightly different modalities of each. So, and in a different disease setting, this time glioblastoma. So, Erica, welcome.
Great. Thank you so much, Bob. Hopefully everybody's seeing my title slide. So thank you so much for your support of our project. And I think I'm very fortunate here to be presenting to this group for the second time. I think it's my second time around as a Buzz Cooper Scholar, which just is such an honor. I can't even tell you to have this opportunity. And so the project that we're focused on, really the question that we're trying to answer is, can we combine novel blood and imaging tests to better monitor patients with glioblastoma? And I'm delighted to present this highly collaborative work on behalf of two of my colleagues, Dr. Steve Bagley on the left-hand side, neuro-oncologist, and Dr. Napovizita on the right, who is a neuroradiologist. And just to put this in the context of the presentation that you just heard, Dr. McCarthy talked about combining two tests to find cancer earlier. Here, we are combining two tests, as Dr. von der Heide said, but this would be after the patient receives a cancer diagnosis and to guide their care. And glioblastoma, as you might already know, is the most common brain tumor in adults, and it's a very stubborn tumor to treat. Tends to occur in younger patients on average than other solid tumors. But to make things worse, monitoring the tumors for these patients can be very challenging. And even for patients who are receiving standard of care therapy, it's very hard to tell if the tumor is getting larger, thus not responding to therapy, or is it shrinking. and actually responding to therapy. And let's go ahead and define and further dimensionalize the problem. Standard of care MRI imaging, so what a patient would usually get, can't always accurately tell us what's happening in the patient's tumor. And so in the setting of glioblastoma, the patient's journey usually begins with surgery to remove as much of the tumor as possible, followed by a combination of radiation and chemotherapy. MRI imaging, so similar to what your orthopedist might order if you have a knee injury or some other kind of sports injury, is actually used to help determine whether or not the patient's tumor is shrinking, as I said, or responding to therapy, or is it in fact growing. However, sometimes the more successful the therapy is in shrinking the tumor, the cloudier the MRI actually looks, making it very difficult to determine what's going on. And we call this phenomenon pseudoprogression because it looks like the tumor is getting larger and progressing, but it's actually getting smaller. And these standard of care images that I'm showing on this slide help to illustrate that problem. I'm showing you in panels A and B the results of standard MRI imaging, and you can see that there's a lot of cloudiness, if you will, in the lower left-hand corner of the image of this patient's brain. When Dr. Nabavizita then followed that up with something called perfusion MRI imaging, the cloudiness seems to go away, suggesting perhaps that this patient's tumor is shrinking, but it's still difficult to tell. So, as I told you, we're going to use two ways to try to address this problem. The first is imaging, and the second is something called liquid biopsy. So on this slide, I'm showing you a novel amino acid PET imaging approach. And here, what we did was enrolled 28 patients in a clinical trial led by Dr. Nabavizita, in which patients received this novel amino acid PET imaging in addition to MRI. And this is really a great example of repurposing because amino acid PET imaging is already routinely used for monitoring of patients who have prostate cancer. And Dr. Nabavizita had the insight to apply this in the setting of GBM. So let me walk you through the results. So the results for our 28 patients are shown in this dot plot on the left-hand side, where we measured something on the y-axis called uptake. And as you can fairly easily see, the patients whose dots are red on the left-hand side who had pseudoprogression, remember, it looks like the tumor is expanding, but it's actually shrinking. Those patients had much lower uptake. Whereas the patients who in the blue dots on the right-hand side who had true progression, their tumors were getting larger, had much higher levels of uptake. And that difference was highly significant at a p-value of 0.009. So what about our patient that I showed you on the prior slide? Using that as a case study, you can see here the MRI images that I already showed you. But as Dr. Nabavizita described to me, this panel D, where we're showing you the amino acid PET results, very clearly shows this bright yellow spot, fairly definitively indicating that this patient's tumor is progressing. And in fact, this patient went on to have surgery where the progression was confirmed. The second thing is liquid biopsy. So we know definitively that tumors, represented here by the blue circles, will shed various materials into the bloodstream. And one of those is called cell-free DNA. It's just naked DNA molecules shed by the tumor. And so my lab has the ability to extract that cell-free DNA out of a tube of patient blood and look at markers called DNA methylation markers. And in a preliminary analysis of a subset of the 28 patients for which we did amino acid PET, I'm showing you a summary of those results here on the right-hand side in a figure that we call a heat map for fairly obvious reasons. So let me walk you through how this is laid out. So in a heat map, blue tends to represent cold regions. In other words, places where there are low or no levels of DNA methylation markers, whereas red tends to represent hotter regions where there are more methylation markers. Very simple to interpret. Each of these columns is a patient. each of these rows is a DNA marker. And it's already fairly apparent that for the patients with pseudoprogression, you can see that the blue markers are much more vivid, if you will, than the corresponding markers for the true progressors. Conversely, you can see at the bottom of the heat map that for the true progressors, these DNA markers are hotter or more red than for the pseudoprogressors. So if you stand back from this heat map, you start to see a very clear signature from the liquid biopsy associating with true progression versus pseudoprogression. So in summary, and as far as our next steps, we will complete the liquid biopsy analysis for the remainder of those 28 patients. We will then take those liquid biopsy results and combine that information algorithmically, probably also with the use of AI, with the amino acid PET readouts from the imaging analysis. We will then take the combined optimal signature and compare that to standard MRI imaging, hoping to display and define a significant improvement in the accuracy. So I thank you very much for your attention, and I welcome your questions.
Thanks, Erica. Similar question that I had for Anne-Marie, these tests, particularly the liquid biopsy test, tell us how routinely available this is now. And if you find out a use for this, is this going to be readily available for patients?
Absolutely. So liquid biopsy in general is routinely available in our clinics. So liquid biopsies are used literally every day to support clinical decision making. And that can sometimes happen in the setting of brain cancer, lung cancer, breast cancer, prostate cancer, and so on. This very highly specialized DNA methylation analysis that I just showed in the heat map, that's something that I would describe as still in the research realm, which is what my laboratory does. However, we work hand in hand with experts in the realm of clinical testing, both in our clinical pathology group as well as with industry partners, such that once a research test is validated, and that would be the next step for our study here, that we can very rapidly develop a plan such that it can be deployed in the clinic and used by physicians. So it's not quite ready for prime time, but if things go well, that would be the outcome of our work.
Yeah, it's like the mirror on your car. Things are closer than they seem.
Yes.
All right, Erica, you're going to go in the standby pavilion, and we'll see you shortly. Our next speaker is Daniel Ackerman. Hi, Dan. How are you? Dan is a research assistant professor in radiology and has been exploring and innovating around metabolic imaging, which he'll tell us about, particularly for liver cancer, yet another very stubborn and difficult to treat tumor. So thanks, Dan.
Yeah, hi, and thank you for having me and for supporting this research. I'll be talking to you about a cancer of the liver called hepatocellular carcinoma, which is one of the few common cancers that is actually increasing in incidence. And as you mentioned, it's an enormous healthcare challenge in our patient population and in the Philadelphia area. And so this is a cancer that, unlike some other cancers, is actually diagnosed using imaging approaches. And treatment can be highly effective if the tumor is small, like you see in this top image here, where it's often treated by surgery. But unfortunately, in most new HCC cases, the tumor has spread beyond this point, and I'm here, and it's very difficult to treat in these cases, which is the majority of cases, unfortunately. So here I'm listing some of the common treatments for this unresectable stage of HCC. And while some patients have a good response from these treatments, overall outcomes are unfortunately quite poor. And one of the reasons is that there's no good way of targeting the right treatment to the right patient. And so these treatments and the sequence of these treatments are selected without any genetic sequencing of the tumor or any such assay. So what we really need is a way to subtype patients before treatment. and then to identify treatments that may be more effective in certain subgroups. And this makes sense because different treatments will target completely different cancer cell vulnerabilities. And so subtyping the patients based on biologically meaningful differences in their tumors could prevent us from prescribing treatments that are unlikely to work. And so an example for that are two common subtypes that are already known, which are those tumors with mutations in the p53 gene or in a gene called beta-catenin or CTNNB1. And evidence is starting to emerge that these two patients in these two subtypes have different sensitivities, or their tumors have different sensitivities to some of the most common treatments. And this is an area of very active investigation. But how do we expand on that, and how can we find the best treatment for every tumor? And so the strength of our lab has been to use biopsies that we've obtained from patients treated at our hospital and generate valuable research models of HCC, which are a powerful way to study cancer biology and a unique resource in the HCC research world. And they've allowed us to perform studies that can't be performed this way without the models. And we've used these models to generate data sets from a collection of tumors that are as diverse as the patient's clinical histories and as diverse as the genetics of these tumors. And we've used them to generate these reliable data sets to study the tumor biology in a number of different ways. And so we've investigated the metabolism of these tumors. And by that, I mean how the tumors process nutrients and drive their growth. And we do this through an approach called metabolomics. We're also able to look at complex patterns in their gene expression patterns. And this term gene expression is a term we use to describe how tumor cells switch genes on and off. And we use an approach called transcriptomics to do that. And then we looked at thousands of circulating proteins, meaning those that we could test for in a blood test, using an approach called proteomics. And the goal of these data sets is to be able to tell you, using unbiased calculations, these are the subtypes of HCC tumors that exist in our data set. And not only that, but using the same data to say, here's the best way to test what subgroup a specific patient is in. And so based on these promising patterns in our data, we've decided to pursue two approaches. The first approach is to focus on these circulating proteins we've identified in our secreted biomarker study. What we found is that genetically distinct tumors secrete completely different sets of proteins into the bloodstream, and this could mean that a simple blood test may be a very effective way to subtype a patient. We're also identifying metabolic differences between patients, and these differences can be quantified non-invasively using functional metabolic imaging, such as the kind that Erica mentioned. And for example, this image here, instead of looking at amino acid uptake, this imaging strategy here looked at glucose uptake, or uptake of a sugar. And this kind of approach will light up some of the patient's cancers in the imaging more than others. And these metabolic signatures tell us something about the biology of their tumors and what treatment may be most appropriate. And so we're excited about developing this further and learning how to apply these biomarkers to HCC treatment. And in practice, this could mean that instead of a one-size-fits-all approach, HCC patients get a blood test and functional metabolic imaging, such as the ones you saw here in the last slide, when they're first diagnosed. And these non-invasive tests would then allow them to be assigned to different subgroups. And on the basis of emerging data, new clinical trials could be initiated, and treatments could be targeted to the subtypes, taking the biology of the cancer into account in selecting these treatments. So I thank you for all your support in this research, and of course, take any questions.
Thanks, Dan. That's very exciting. And just to clarify, this is precision oncology, where we treat patients according to the type of liver cancer they have. None of what you're proposing involves having to stick a needle in the liver and biopsy it. This is all that,
right, and that's
which is different than our current approach.
Right, that's been one of the difficulties in introducing precision medicine to HCC, is that HCC treatment is that a lot of patients are never biopsied because the diagnosis is made based on imaging findings. And so trying to introduce a more targeted approach requires a non-invasive strategy.
Yeah, that's great. Okay, we'll see you also in a few minutes, and we have one final investigator to introduce their work, and that's Andres Blanco. So who will come online here just now. Andres is assistant professor in biomedical services at the University of Pennsylvania School of Veterinary Medicine. I'll just emphasize that point, that the Abramson Cancer Center is across many schools, not just the School of Medicine, and Andres is at the vet school. Vet school actually has a cancer center for privately owned animals, many of whom develop cancer. Andres is working in an area that is relevant to all patients with cancer. So, and that being colon cancer. So Andres, welcome, thank you.
Okay, thanks very much, Bob, and thanks so much for the opportunity to speak here. I'm thrilled to be able to tell you about the project we've been doing with ACC funding. So we are studying the process of cancer metastasis. That's the spread of cancer cells from a localized primary tumor to a distant organ, a distant part of the body. And as you may well know, metastasis is the real killer in cancer, in most cancers. As shown in colorectal cancer, what we're working on in this project, if the patient is diagnosed with disease when it's localized in the primary tumor stage, prognosis is good. But if metastasis has already occurred, prognosis is very poor. So what really is metastasis? How does this process work? Well, simply put, it starts with, of course, an original primary tumor, which is an area of uncontrolled cellular growth that grows, again, for the most part, locally in a localized physical tumor until something happens. Something switches in a cell or many of the cells within the primary tumor that allows them to move and break off of that tumor and invade into the bloodstream, which gives them access to many different distant organs. And by colonizing distant organs, that's metastasis, that compromises organ functionality, and ultimately that's usually what patients die of in cancer. So how do we think about it? Well, we like to go zoomed way in and think about this on the level of single cells. When we think about single cells in cancer progression, well, we know that cancers start really from one cell that has had some genetic mutations that make it grow uncontrollably. And we know that one cell becomes two cells become four become eight, and so on. What we don't know is how and when and where in this tumor this switch happens, such that some cells, or maybe it's many different cells in the tumor, gain this ability to invade and become metastatic. And once they do that, are all the metastatic cells the same, or do they continue to get maybe more and more aggressive? So in thinking about this, what we're looking at here made us realize, there are ways to analyze this type of problem from other fields, one of which would be to use an evolutionary phylogeny, which has been extremely helpful for understanding speciation events in evolutionary biology. And these types of analyses are very helpful for understanding how closely species are related, not just to each other, but what is their shared ancestry. Did they come from a common ancestor, or did they evolve, multiple times independent paths to the same outcome? We thought if we could do something like make a cancer cell phylogeny of our cells rather than species, that could be very powerful to understand metastasis. So we sought out to do just that. We made a system, we call it Max Gestalt, and this is a system where we rig this amazing methodology called CRISPR-Cas9. You may have heard of this, just recently won the Nobel Prize. We rig it such that we have cancer cells that ultimately we're going to be able to inject into mice to have this CRISPR-Cas9 system insert a little tiny bit of DNA, tiny little stretch of DNA into the cells, which makes what we call a barcode. It doesn't affect anything in the functionality of the cells. It's just a little bit of a tag we put into the genome to make that genome, to make that cell identifiable when we, at the end of the day, do genome sequencing on the whole population of cells. Now, it's not only that, but when the cells divide, each daughter cell from a parent both maintains the original barcode of DNA from the parent, but also gets a new and different barcode that now makes this cell uniquely identifiable from this one and from this one. And over time, again, 2 cells become 4 become 8. Eventually, we have millions and even billions of cells. And when we harvest the cancer cells from the mice, the primary tumor, as well as all the different metastases that we have, and we do a lot of computational and mathematical analyses of these barcodes, we can eventually reconstruct what we call a cancer cell phylogeny. Now, I know this part looks very abstract, but through analyses of data like this and related ways of looking at the data, we can actually start to get a lot of information about the patterns of spread. Just for example, this area here, this represents in a kind of abstract way cells that have remained in the primary tumor and branching events that lead to cell doublings, but that still remain localized. Whereas these regions over here, different blobs, represent events that have led to metastatic spread to different organs. And we can start to ask things like, are these cells, this metastasis come from cells with a primary tumor? Did they come from cells in another metastasis? How aggressive are these different metastatic cells? So on and so forth. Ultimately, at the end of the day, through analyzing these data, we've been able to identify genes that we think are actually helping to promote the metastatic process, to help drive metastasis. And we're now testing that hypothesis by taking some of these genes and implanting colorectal cancer cells into a mouse, either regular cells or cells where we delete a gene of interest, and then followed by imaging the cancer cells over time. And what we're seeing is that for some of the genes we're interested in, we find that if we've deleted that gene, the mice do not go on to get metastasis over time, and they live much longer. Ultimately, where we're going with this at the end of the day, we can't just delete a gene in a patient's cancer cells, at least not yet. But some of the genes that we're identifying may have small molecule inhibitors, or we may be able to develop those inhibitors such that eventually in mice and then at some point maybe in humans, we would have a drug where we can treat the patient and have that drug block and ideally eliminate metastatic spread so that the patient can have extended lifespan or cured. So that is our project. I'd be happy to take any questions.
Okay, that's awesome. And the notion, let's say you had that drug, these would most likely be given to maybe patients who we're worried about after their surgery. We're worried there's lurking cells, and you would block the ability of those cells to grow and be metastatic. Is that the idea?
Yeah, that's definitely the idea. Again, it depends also on what stage the patient's disease might be caught at. If the tumor is caught at a stage where it can be resected and they can remove the primary tumor, and as you say, there might be residual metastatic cells, that would be a great application for such a drug in principle. And depending on how the exact activity of the gene and what stage of metastasis it's actually helping to drive, it could be that even if the patients have inoperable tumors, if this drug is really blocking metastatic progression, maybe it could be treated to help eliminate the metastases, even after they've been established. But it remains to be seen ultimately how we'll be able to utilize such drugs.
Well, that is very exciting. And okay, so thank you. Let's bring everyone in from the waiting area. And while folks are joining, I'll just say for the audience, what we'd love to hear from you, and you can put your questions in the chat, and Carolyn Brown is here to help us read them out. Taking a step back from today, I think it's important to point out, what are we doing here? We're studying some of the toughest problems in cancer. They are manifesting in multiple cancer types: breast cancer, brain cancer, liver cancer, colon cancer, and we're searching for new approaches that have new synergies that will allow us to be precise in how we treat. Really, cancer is moving away from one size fits all, but what kind of cancer does this patient have and how will we treat it? And that's how we're going to improve quality of care and outcome. So there is our distinguished committee, as you can see there. We're going to go by first names, if that's okay. And Carolyn, I think some questions have come in, so I'll have you ask.
Yes, thank you, Dr. Van der Heijden, and thank you to our fantastic panelists for sharing your research updates with us today. Our first question is, how do you measure success?
Okay, well, maybe everyone can give a sentence or as to like what are your metrics of success, or what do you hope to achieve? Maybe we'll start with Dan.
Yeah, that's really interesting. For me, it's one of the things I always think about is I work with a lot of physicians that are, on the front lines treating these cases, and when I present the work, how excited do they get? Because
Yeah.
sometimes I can get excited about a new biological mechanism I think I'm on the track of, or some feature of the data that I think is interesting. But the people that are really treating the cancer, they're often aware of some nuances in how the cancer is being treated and what some of the limitations are that really change how I would prioritize it. So for me, that would definitely be a way I measure the success or potential success of an idea.
Yeah. Anne-Marie, what would you add?
Yeah, I think that for me, I got into this field because of, my own personal experience with cancer in my family. And so for me, I measure success in, is my work inching us closer to helping patients avoid cancer or catch cancer at its earliest phase. And so one of the reasons I love being an epidemiologist at Penn is because I'm surrounded by so many clinicians that are helping me to do work that is vital to patients.
Yeah, that was a clear theme from all of you, how your questions you're asking in the lab are informed by your clinical colleagues because you're talking to them. Erica, could you answer the success question? And then I have a specific question for Andre.
You bet. So in our case, we have a very clear question, right? The question is, can we combine liquid biopsy and this new imaging to improve what we're already doing with MRI? So MRI is our benchmark. And if we can demonstrate that we have a significantly better accuracy of discerning pseudoprogression from true progression, that would be our first measure of success. But I do want to circle back to the question that you asked me, Bob, a few minutes ago, which is this in the clinic? And I would say that's our second measure of success. So if we can complete the study and demonstrate better accuracy, if we can then validate that result in an independent, larger cohort and pave the way to get this into the clinic, that's success.
Yeah, it's I love that thought. We've often thought about, what is success? Success is if clinicians use these ideas, if it adds value. It's exactly what Dan was saying. Do they get excited when they hear this? That's our North Star. Is it helping patients? Andres, Chris Hall has a question for you. And this notion of targeting genes that drive metastasis, do we presently have that ability in any way?
Right. Yeah, that's a great question. It's on the map, and there are some drugs that have cases in a few in clinical trials that are thought to be primarily acting through targeting metastasis, but it's been very difficult to find drugs that really are, most effective in the later stages of cell growth and the cellular spread stage. So still, by and large, the vast majority of drugs are just sort of general anti-cancer. They might be very, in a way, personalized, as we say, to that specific type of cancer, or even that patient's, the genetic mutations in that patient's cancer, but they're not usually focused to this sort of like particular later stage. And so there are, a lot of people who are still trying to figure out what are the right targets that would be most helpful for treating the later stages. So there's a little bit out there, but not too much.
Yeah, it's target identification, and that's exactly what your CRISPR screen is about. We have a question from Charles Zen. Hi, Charles. And it's directed to Dan, but maybe others can jump in. Dan, it's my understanding, this is from Charles, that it now costs $1,000 to sequence an entire human genome, and that figure will be just a few $100, $300 in a few years. Why not just sequence the whole genome of a patient, identify the mutations you're looking for? You look like you're keen to answer.
Absolutely. So in the case of a tumor, of course, the tumor now has a different genome than the person, right? It's acquired all these mutations that drive tumorigenesis, and so in some cancers, it's very easy to get a piece of that tumor to sequence, and where that's useful, people do that already. But in HCC, we just these tumors are never biopsied, so we don't actually get a piece of that tissue to do sequencing on. And the other problem is, even if we did that additional procedure and got a piece of tissue and sequenced it, there's currently not a lot of information on what mutations are targetable in HCC. Only some mutations in some cancers are actually targetable through a targeted therapy. And so it gets back to this issue of subtyping, really. What subtypes are informative for informing therapy? And you know, another aspect can be that different mutations can actually have similar consequences in terms of the metabolic changes, for example, that we could see through imaging. The cell can get to a similar highly proliferative state using different mutations, and so sometimes even mutations aren't the whole story. So I think that what we're trying to do is really understand the entire biology of this cancer. And in HCC especially, I've been focused on trying to understand that, figure out ways to understand that without another invasive test, but by doing a non-invasive. So it's a very good question, and I think that in the future, maybe more patients will be biopsied, but for those that aren't biopsied, we do need a.
Yeah, it's so just to add a couple of things to that. Measuring the genome that we're born with and measuring the genomics of the tumor can be quite a bit different, and there are opportunities on both sides. Initially, most of the targeted therapies were based on genes in the tumor, but now we're learning how inherited genes can be targeted. BRCA1 and 2 is a great example of that. Anne-Marie works on that. Yes, we have a huge advantage that cost of sequencing is coming down. I would just add that that's becoming the easy part and the cheap part, and the bioinformatics is the crazy part. And the Breakthrough Challenge probably remembers last year, the year before, we funded bioinformatics as our innovation specialty. Comment from Jeff Krieger: very impressive presentations that'll motivate the board, our writers, sponsors, donors to contribute funding for future similar research projects. Thank you, Jeff. Good to hear from you. A question, maybe we'll see who wants to answer. Emily Whitehead, who received CART19, just celebrated 10 years of being cancer-free, and that was all over Twitter and in the news. She has grown up to be an adult now, which is remarkable, as have thousands of children treated now with that gene therapy. Where does gene therapy go from here in cancer? Which that's a question that has no wrong answer. So, who on the panel would like to add a thought or two?
Bob, I could speak to gene therapy and brain cancer. So, you know, Emily Whitehead, as probably everybody in the room knows, suffered from a blood cancer where the tumor cells are circulating around in the blood, in the periphery. And one of the things that Penn has been a real leader of just in the last few years, I mean, this is very new, is trying to harness the power of gene therapy for solid tumors. So a solid tumor sits in one place. It may metastasize, as Andres described, but for the most part, it's stationary and it's large, and characteristics of the tumor change as you get deeper into that solid mass. So it presents a number of problems that are different than a hematologic malignancy like Emily had. And so that's what we're trying to tackle in our neuro-oncology neurosurgery team for glioblastoma. And so we've had a couple of very important clinical trials led by my colleague Don O'Rourke, neurosurgeon. Those have brought some success in understanding the biology of how these CAR-T and these gene therapies work in the setting of the brain. We're still working out the details, And importantly, if you remember the imaging and liquid biopsy that I talked about just for managing standard of care patients, all of that work is also embedded into the CAR T clinical trials. So that once we get the CAR T therapy working in the brain and for patients with GBM, our hope is we're standing ready with these biomarkers that will help us figure out when the patients are responding and when they're not, and so on and so forth. So stay tuned. I think you're going to hear a lot in the next couple of years, and we are just standing up our next CAR T trial as we speak. I think we'll start enrollment on that actually very shortly. So
I would just add a perspective that's a 20-year perspective. I love the question because it's like, what can gene therapy do next? But if you go back 20 years, the field was in great disarray with some terrible outcomes, and Penn was, at the leading edge of that, as we all know. And now here we have the first ever cancer gene therapy approved in the United States, done here by work at Penn, and we're looking for more. I mean, I remember gene therapy protocols 15 years ago where I couldn't find, at some institutions, the IRB would not accept any consideration of a gene therapy approach. So what's happened? Well, we've learned a lot about how to be very precise in our techniques, techniques that didn't exist before. We're learning how to do cell manipulation outside of the body, so the gene therapy actually happens in a controlled environment, not only for T cells, but also for stem cells. We've just about to start a transplant trial with gene therapy. And what's really remarkable is that it's happening here in Philadelphia. With cell and gene therapy, the industry that's grown up around Penn technology is amazing. Some people call it Silicon Valley. More than $1 billion of VC money now invested, and companies pouring in, really making this real. Penn has more patents in cell and gene therapy than any other institution in the world. And we've seen remarkable features and outcomes also in solving genetic diseases, particularly rare diseases, and our partners at CHOP have been remarkable in that. So there's so much now, and we've learned so much that it's like a whole new way of medicine. Bill Lipscomb has a question. Dr. McCarthy, Anne-Marie, we're going to go by first. How are you recruiting Black women to participate in this study? What are your approaches for that?
Yeah, so for my work, I'm primarily right now building cohorts from our electronic health record data. So I've developed a cohort with the help of my colleagues of all women who come through Penn Medicine for a mammogram. And then we've linked that data with outcomes. So we've linked to the cancer registries to determine whether women have developed cancer over time. And so we can use the data that we have to start generating some of our biomarkers and seeing how well they predict risk. I am also doing some work that's funded by the ACC pilot grant and some other funds to try to increase the enrollment of Black women in the Penn Medicine Biobank, because as we're rolling out the biobank, it just takes time for patients to join, and so we are reaching out to patients using the Penn Medicine EHR and through email to invite women, specifically Black women who have had breast cancer, to join the Penn Medicine. And we are hoping that by doing that, we're going to be able to expedite our research and have the large numbers of Black women in our studies to be able to make some conclusion. And so right now we're just working on developing our risk markers. And once we get that data and see how well they work, then we can start moving to more interventional work. And for that, we'll need to actively recruit patients. So the support of the pilot grant is really critical because it allows us to generate the preliminary data that will help us design our future studies that actually help to translate into the clinical space.
It's been a really high priority for the Abramson Cancer Center to make sure all our studies reach all patient populations, and really a decade of work, much of it led by one of our leaders, Carmen Guerra, to form bidirectional relationships with all communities has really shown success, where unlike in other situations or other parts of the country, the percentage of Black participants on our clinical trials actually matches or exceeds the percentage of Black individuals who live in the service area, our catchment area around Philadelphia. And so it doesn't just happen. We have to be very active and thinking about obstacles and barriers that for enrollment that impact one group or another. Individuals who join clinical trials, often the visits are more frequent. There's costs associated with that. They have babysitting, time off work, travel expenses, and we now have a program for those who have a financial need to, if they join a clinical trial, we are partnering with Lazarus Foundation to pay those incidental costs. That was initially concerned that would be considered inducement, and we worked with the state of Pennsylvania and the governor to pass a law saying that in fact was not inducement, and that's really changed things. So lots of effort in just so many ways, but it comes down to, as Anne Marie said, the investigators realizing there has to be particular outreach, and it has to be an active effort. Bill Barber, thanks for your comment. Excited to generate more funding. I'm going to ask Carolyn if we have time. No, it's 1:58. We should thank our sponsors. Do we have the sponsor slide that we can put up? Look at these amazing people. Wouldn't be possible without all of you, and of course to the board and for the Breakthrough Challenge. We are super excited to be out there biking and raising money and bringing it back and doing more discovery research. So September 18th, that's our big day. Carolyn, did you want to say anything to everyone before we go?
Thank you again for joining us today. Thank you to our panelists and to Dr. Vonderheide. I would be remiss if I did not also thank the Breakthrough Challenge Board of Directors and the Zent Family Foundation for your continued support. We hope to see you all on September 18th, and we have a virtual event September 5th through September 18th if you cannot make it in person. Thank you again, and take care, everyone.